Hello folks, my name is Logan Thrasher Collins, and I'm going to tell you about some really exciting nanotechnology research from the Baker Laboratory at the University of Washington, Seattle. Specifically, we're going to be learning about 2D sheet-like lattices constructed from a pair of de novo engineered proteins. The research that I'm going to discuss was published in the scientific journal Nature during 2021. Here's the title of the paper for those of you who are interested in looking it up and learning more. So, what is de novo protein engineering? It is a way of designing new proteins from scratch, guided by desired function and biophysical constraints. An example of a biophysical constraint is that if different parts of the designed protein cannot sterically clash, which means that atoms cannot physically overlap or come too close to each other. De novo protein engineering often involves first specifying a three-dimensional shape for the protein and then using computational methods to find an amino acid sequence which folds into that shape. The computational methods can involve searching for and linking together small fragments of known proteins which might help achieve the desired shape, as well as performing physics-based modeling. Another common part of de novo protein engineering is using various computational methods to design interfaces between two synthetic proteins which allow those proteins to stick together. The 2021 paper on 2D protein lattices advanced the field of protein engineering by employing de novo protein design methods to create two-component hexagonal protein lattices. Two proteins, hereafter referred to as A and B, were computationally reconstructed from de novo building blocks developed by the Baker Lab. The software Rosetta, which was also developed by the Baker Lab, was used to design a specific A-B binding interface. In addition, an AGFP version of A was made as a fusion protein. Importantly, the GFP was placed in a location which did not block the A-B interface. The type of GFP used was an especially stable form of GFP called superfolder GFP or SFGFP, but I will just call it GFP from here on out. To make their computational designs a reality, the Baker group first co-expressed AGFP and B in E. coli. They also co-expressed A and B in E. coli as a comparison to make sure that the AGFP version worked just as well as the A without GFP. Here you can see a fluorescence image of E. coli co-expressing AGFP and B. Because the two proteins assemble into hexagonal lattices, they localize to spots within the bacteria rather than being evenly distributed throughout the cells. You can also see transmission electron micrographs of purified lattices made by E. coli. In the close-up of the AGFP plus B lattice, you can see the GFP as little bumps at the vertices of the hexagons. The group also expressed each protein individually in bacteria, purified them, and then assembled them into lattices in vitro. When AGFP was stored at room temperature, no aggregation was observed even after several months. The same lack of aggregation happened when storing B. One great thing about these lattices is that their assembly can be induced upon mixing of the components. The sheets of AGFP and B, which were made in vitro, were of an even higher quality than those made inside of bacteria. You can see how clean the lattice is in this negative stain transmission electron micrograph. Some other excellent features of these lattices include their rapid concentration-dependent growth and their ability to self-heal. The rate of growth can be increased by increasing the concentrations of AGFP and B. Defects and vacancies in the 2D crystal lattice get fixed over time because the lattice spontaneously reassembles into the most stable state, and free-floating soluble AGFP and B are available to fill any openings. You can see these properties illustrated by atomic force microscopy. The lattice grows quickly and heals quickly. Next, the lattices were stuck onto cultured cells using a synthetic receptor. The synthetic receptor was a fusion protein with an extracellular anti-GFP nanobody to bind AGFP, a transmembrane domain to anchor it into the membrane, and an intracellular M-scarlet fluorescent protein. These synthetic receptors were expressed on the membranes of cultured cells, and the protein lattices were applied. The locations where the lattices had bound the membrane were visualized using the co-localization of GFP fluorescence and M-scarlet fluorescence. It was also important to show that the lattices could induce cell signaling by clustering some kind of naturally occurring receptor. The authors of the paper chose to target the Tie2 receptor, which is involved in blood vessel growth. They employed SpyTag and SpyCatcher to link a Tie2 ligand, known as Ang1, onto the A protein component. SpyTag and SpyCatcher are a pair of small proteins which spontaneously form a covalent bond to each other when mixed together. Preformed lattices equipped with the ligand Ang1 were added to cultured cells. These lattices stuck onto cell membranes through the interaction between Ang1 and the Tie2 receptor. This triggered clustering of Tie2 receptors, which led to strong induction of cell signaling pathways. As a result, the actin cytoskeleton underwent remodeling in the affected cells. You can see the cytoskeletal remodeling here in these 3D reconstructions from super-resolution microscopy images. Finally, 2D protein lattices were assembled directly on cell membranes. To do this, a BGFP protein, similar to AGFP, was made and added to cells expressing the GFP binding synthetic receptor described earlier. The BGFP was bound to the synthetic receptors, yet did not form a lattice on its own. Next, the A protein was added to the cell culture. This triggered lattice assembly on the cell membranes. You can see it happening in these fluorescence microscopy images. With BGFP only, there are tiny specks of green, indicating the binding of BGFP to the synthetic receptor. Upon the addition of A, the specks grow into much larger spots, showing that the lattices are assembling on the membranes. An interesting property of the lattices was that they inhibited endocytosis, likely because the cell membrane would have difficulty invaginating when the lattices are stuck to it. This could be useful for cell signaling applications, since endocytosis often removes receptors from the membrane after too much stimulation. By preventing endocytosis, more sustained signaling might be achieved. So why is this useful and important? Firstly, the two-component nature of these lattices allows assembly to be induced in a highly controllable fashion, which is an advance compared to past research. There are many possible applications for 2D protein lattices, but as I've discussed, their capacity for strongly inducing cell signaling through receptor clustering will be especially useful. The lattices could be applied as a new kind of medicine, which circumvents many of the issues associated with small molecule drugs. In particular, many of the cells in the immune system need something to cluster their receptors in order to activate them. 2D protein lattices could be used to induce strong immune cell activation in situations where it is needed, such as when someone has cancer. But this is just one example. There exists a vast array of possibilities for applying 2D protein lattices to biological challenges. Thank you for listening. I hope you enjoyed my discussion of this exciting research.